You are reading about mitosis and meiosis, cell division, and you're reading it like a court case. So understand some of the characters and what they're doing. All right, it's going to make a lot more sense if you have a good idea of who the characters are. So, for example, live is a liver cell, that's a body cell. The defendant is an oocyte, that's the person that's on trial. A nuclear is going to be a skin cell that's going to discuss mitosis specifically. And then a sperm cell is going to discuss meiosis because sperm cells and egg cells, gametes, undergo meiosis, whereas skin cells, body cells, liver cells undergo mitosis. So here's a little concept map to sort of see some of the terms that would be associated with mitosis as opposed to meiosis. All right, so this is all under the umbrella of cell division. Apoptosis just means programmed cell death. If something goes wrong with the cell division cycle, then the cell's programmed to self-destruct. That way, it's less likely to cause a major problem, like cancer, for example, uncontrolled cell division. A cytokinesis has to do with dividing everything other than the nucleus and the nuclear material. So mitosis and meiosis are mostly going to be about dividing up the chromosomes and all the DNA, making sure that's even. Cytokinesis, on the other hand, is going to make sure it's dividing up the organelles. All right, so most of this process is going to have to do with dividing up of the DNA. Haploid means half. So I'm on the meiosis side here, and haploid means half the total number of chromosomes. So we normally have 46 chromosomes, half would be 23. That's the diploid number, 46, the double number. Homologous just means those chromosomes are the same. They're the same size, same shape, they have the same genetic material. Non-disjunction is sometimes those chromosomes, they don't separate and go to two different cells. Instead, they stay together, and as a result, you get an extra chromosome in the mix. On the mitosis side, mutagens are cancer-causing agents, carcinogens, mutagens. Something has been mutated in the DNA. Somatic just refers to body cells. A sister chromatin is a way of describing a duplicated chromosome. So DNA goes through the process of replication, it duplicates itself or replicates itself, and now we have these two copies that are together. A binary fission is also a type of mitosis, but this has to do with things like bacterial cells. So here's an example of a bacterial cell. Duplicates its DNA, it divides into separate daughter cells. That's binary fission. So these are single-celled unicellular organisms. This is how they divide. The cell cycle is more than just mitosis, cell division. Most of the cell cycle is going to be interphase. During interphase, we have this G1 where it's basically growth. So G just stands for growth, and the cell's just growing in size. Then there's a checkpoint there. If the cell is large enough, it can move to the S phase. S phase stands for synthesis. We're synthesizing, making more DNA. After the DNA is copied, there's another checkpoint just to make sure that the cell's the right size and that the DNA is copied correctly. Then it goes through the stages of mitosis. You'll notice that the stages of mitosis are a very small part of the cell's life cycle. In fact, many cells never even make it to mitosis. Their whole life is spent in interphase. For example, muscle cells, nerve cells, they don't divide. So during mitosis, there's one more checkpoint just to make sure everything is happening normally. And that happens pretty early on. And the idea there is just to make sure that each new cell has an equal amount of DNA. If any of these checkpoints fail, then the cell goes to apoptosis, that is self-destructs. All right, what are the phases then of mitosis? So the easy way to remember this is IPMAT, I-P-M-A-T, IPMAT. Interphase is first. then prophase, metaphase, anaphase, telophase. IPMAT, IPMAT. So interphase, just normal cell life where it's growing and organelles are just doing their normal daily jobs. But once we get to this S phase and G2 phase, then the cell is going to go through mitosis. It's going to divide. When it divides, the key is dividing all of the nuclear material. It's also important to make sure there's enough of the organelles divided between the two new cells so they can survive, which is why it's important that the cell gets a chance to grow large enough, but not too large. If a cell grows too large, then again, it won't survive. It's too big. After interphase, when cell division starts, it enters prophase. So during prophase, the nucleus begins to disappear and the chromosomes begin to condense so that they become more visible. Like rather than just being this big blob of DNA, the X's, the chromosomes kind of have this X kind of shape. They become more visible. Prophase. During metaphase, all the chromosomes line up in the middle. So you can remember metaphase, middle. Anaphase, the chromosomes start moving toward opposite sides. Anaphase, away. Think of the word away. And then telophase, we've got two new nuclei. So think of the word two. Telophase, two new nuclei. I think of it kind of like gym class. Prophase is just when the class begins. So everyone first comes to the gym and they're just all milling around. But then the teachers come out, they blow their whistle, and the classes start to form. You start to see that there's different classes all in the same gym. So that reminds me of prophase. The chromosomes start to condense. The students are like the chromosomes. Then the teachers say, we're going to play kickball, volleyball, dodgeball, whatever it is. And in order to do that, we're going to pick teams. So the two team captains, they are the centrioles of the cell. So they go to either side. Everyone else lines up in the middle. That would be like metaphase. Then the captains start picking, and they're trying to pick the teams evenly. And as they pick the teams, those people start moving toward them, just like the chromosomes move toward the centrioles. So it would be anaphase. They're moving away from the middle and toward the captains. Then, once everyone's picked, it's telophase. You've got two new teams. Just like in mitosis, you have two new nuclei. Cytokinesis then divides everything else, and we get two new cells.